I was just reading the latest clinical data on autophagy, and it changes how we should evaluate cellular recycling, because the way most people talk about this process has almost nothing to do with the actual biology. The wellness industry has turned autophagy into a fasting benefit. Skip meals and your cells magically clean themselves. It sounds simple, actionable, and empowering. It is also dangerously incomplete. Autophagy is not a button you press by missing breakfast. It is a tightly regulated biological system with specific molecular machinery, multiple regulatory inputs, and consequences that run in both directions. The question is not whether fasting influences autophagy. The question is whether the simplified narrative that people are following actually matches the biological reality of what this system does. Now, when you examine autophagy through a cellular mechanism lens, the story becomes more complex, more conditional, and far more consequential than any trending protocol has suggested. The real question is not how to activate autophagy. It is what happens when this recycling system becomes dysregulated, and why that distinction matters more than the activation story itself. The popular claim is straightforward. Restrict your eating window, and your cells enter a detox mode. This framing has shaped how many people approach fasting without any mechanistic qualification. But this framing confuses activation with regulation. Autophagy is the major intracellular degradation system. It is not synonymous with detoxification, and the difference is not trivial. When autophagy is described as a detox, the critical distinctions between degradation, recycling, and simple waste removal collapse into a single misleading concept that obscures the actual biology. The consequence is that people pursue fasting protocols with confidence built on a misunderstanding, believing they are detoxifying when they are engaging a system they have not been taught to evaluate. Here is the mechanistic question. What happens when autophagy is not properly regulated? Because insufficient autophagy and excessive autophagy both carry distinct and measurable biological consequences that the trending narrative ignores. Understanding those consequences requires separating what autophagy actually does at the cellular level from what the wellness narrative claims it does at the level of lifestyle and protocol. Autophagy, derived from the Greek for self-eating, refers to the major intracellular degradation system by which cytoplasmic materials, including damaged organelles and misfolded proteins, are delivered to and degraded in the lysosomes. This is not a vague cleansing event. It is a specific molecular process that produces new building blocks and energy for cellular renovation and homeostasis, recycling at the most fundamental biological level. The lysosome functions as the processing center. It receives cellular cargo, breaks it down into amino acids, lipids, and nucleotides, and returns those components for immediate reuse in new cellular constructions. The discovery of the genes governing this process earned Yoshinori Ohsumi the 2016 Nobel Prize in Physiology or Medicine, recognizing decades of work on the molecular machinery that makes autophagy possible. Ohsumi identified the ATG genes, autophagy-related genes, in yeast, establishing for the first time the genetic blueprint that cells use to execute this recycling program. These discoveries moved autophagy from an observed phenomenon to a genetically characterized process, confirming it as a fundamental feature of cellular life rather than a modern wellness invention. But understanding the molecular machinery is different from understanding when and how it activates. The presence of the system does not tell you how it is regulated or when it should engage. The critical distinction that the detox narrative misses is this. Autophagy is regulated, meaning its activity level, its timing, and its tissue-specific behavior all matter as much as its existence. When nutrients become scarce, cells face a fundamental resource allocation problem. They must determine what to preserve, what to degrade, and how to redistribute limited molecular resources to sustain function. Autophagy is a major mechanism by which nutrient-starved cells reallocate resources from less essential to more essential processes. This is survival logic at the cellular level, not a cleansing ritual. The cell is not flushing toxins. It is making calculated biological trade-offs, degrading dispensable components to generate the raw materials needed to maintain its most critical operations and structures. So the lysosome receives the cellular cargo and executes precise enzymatic degradation, breaking down proteins, lipids, and organelle fragments into reusable molecular components that the cell can immediately repurpose. This is what makes autophagy a renovation system rather than a waste disposal system. The outputs of degradation become the inputs for new construction, a closed-loop recycling process at the molecular scale. Intermittent fasting has been studied as a modulator of autophagic activity. But modulation is a precise term in this context, and it does not mean the same thing as reliable activation. The relationship between fasting duration, fasting intensity, and the degree of autophagic response is not linear. It varies by tissue type, metabolic state, and individual cellular context. Short periods of nutrient restriction may initiate autophagic signaling, but the magnitude and duration of the response depend on which cells are affected and what regulatory signals are present. Different tissues respond to nutrient deprivation at different rates and through different regulatory checkpoints. Liver cells, muscle cells, and neurons do not activate autophagy on the same schedule or threshold. A nutrient-sensing pathway called mTOR acts as the primary regulator in this system. When nutrients are abundant, mTOR suppresses autophagy. When nutrient availability declines, that suppression is released and recycling increases. This creates a regulated balance between growth and recycling, not a binary switch you flip, but a spectrum of activity governed by multiple upstream signals and cellular conditions. When autophagy is insufficient, damaged organelles and misfolded proteins begin to accumulate within the cell. Over time, this progressive accumulation can contribute to cellular dysfunction and reduce tissue performance. When autophagy is excessive or dysregulated, the cell may begin degrading essential components that it needs to survive, effectively dismantling its own operational infrastructure in an uncontrolled recycling cascade. This dual risk, too little recycling and too much recycling, is precisely what the simplified fasting narrative fails to communicate. Both directions of dysregulation carry real biological consequences. The biological reality is a carefully calibrated system in which context, timing, nutrient status, and cellular demand collectively determine whether autophagic activity serves the cell or begins to undermine it. The important limitation is this. Much of the foundational autophagy research, including Osumi's Nobel-recognized work, was conducted in yeast models, organisms with fundamentally different cellular complexity than humans. Translating findings from yeast and animal models directly to human health protocols requires a degree of caution that the wellness conversation around autophagy has consistently failed to exercise. Animal studies on fasting-induced autophagy may not directly predict human responses. Differences in metabolic rate, tissue composition, and fasting tolerance create meaningful gaps between model organisms and clinical application. There are specific populations for whom aggressive fasting protocols may be counterproductive or harmful, including individuals with metabolic conditions, those with a history of disordered eating, or people dependent on medication timing. What does hold up is the concept itself. Autophagy as a cellular maintenance and recycling system is an established biological process, confirmed by decades of molecular research and genetic characterization. What does not hold up is the claim that a specific fasting window reliably optimizes autophagy for a predictable health outcome without accounting for individual variation and clinical context. The mechanism is real and well characterized at the molecular level. The simplified protocol built on top of it is the problem. And that distinction is exactly where the conversation needs correction. So does fasting really detox your cells? The honest answer is that the question itself contains the core problem. The word detox does not describe what autophagy actually does. Autophagy is a genuine and essential cellular recycling system. Cytoplasmic materials are delivered to the lysosome, degraded and repurposed. That is not detoxification. That is biological renovation. Nutrient restriction can modulate autophagic activity. That relationship has been observed under controlled experimental conditions, but modulation under controlled conditions is not a guarantee of a specific health outcome. Calling this process a detox misrepresents both the mechanism and the degree of control that any dietary protocol provides over a system regulated by multiple independent molecular inputs. Autophagy does not flush toxins from the cell. It degrades and recycles damaged cellular components through lysosomal pathways, a distinction that fundamentally changes what you should realistically expect. The difference between recycling and detoxification is not semantic. It changes the questions you should ask, the outcomes you should anticipate, and the protocols you should be willing to evaluate. Expecting a detox leads to oversimplified protocols and unearned confidence. Understanding a regulated recycling system leads to better questions and more honest evaluation of what fasting can achieve. The cellular machinery identified by decades of research and recognized by a Nobel Prize operates with a molecular precision that no trending dietary protocol can fully replicate or reliably replace. The resolution is not that autophagy is irrelevant to health. It is that this mechanism deserves a far more rigorous and more honest conversation than the one it has currently been given. If you want to evaluate the claims being made about fasting and cellular health, the starting point is separating the verified biological mechanism from the marketing narrative built around it. Ask what the specific biological process is before adopting any protocol that claims to activate it. The mechanism should be identifiable and supported, not assumed or marketed. Recognize that autophagy operates on a regulated spectrum influenced by multiple cellular inputs. It is not a switch you can reliably control with a meal schedule alone. Consider that tissue-specific responses, individual metabolic context, and upstream regulatory pathways all influence whether a given fasting protocol produces any meaningful autophagic effect. When evaluating health claims about fasting, look for whether the claim specifies the biological mechanism, the study model, the study population, and the limitations of the cited evidence. This episode is educational interpretation of established biological concepts and supplied research findings. It is not individualized medical advice. Any significant dietary change warrants discussion with a qualified professional. The goal is not to dismiss autophagy or discourage interest in fasting. It is to respect the biology enough to understand it accurately before applying it to your own decisions. Cellular maintenance is not a trend. It is a fundamental biological process that predates every wellness brand that has borrowed its name and simplified its function. The biology is precise, the mechanism is real, and the conversation deserves to match that precision. Optimize the protocol, protect your brain, and I'll see you in the next data set.